Assalamualaikum, my name is Nur Syahira binti Muhammad Sijihan, and for my FIP project title is a systematic review on biosynthesis, bioengineering and sustainable productions of microsporin like amino acids or MAAs, and I'm supervised by Puan Rafidah binti Rasul. Let's start with an introduction. So firstly, for the background of study, so what is MAAs or microsporin like amino acids? So MAAs are a class of natural UV absorbing compounds with a broad spectrum of biological functions. So in recent years, natural bioactive compounds have gained attention because they can be used in various of fields. And among of these bioactive compounds, natural bioactive compounds, MAAs are studied for their unique properties. And it can be found in aquatic habitat organism, in which important for shielding aquatic life from harmful UV radiation. So MAAs have various bioactivities such as potential anti-aging effects and also photoprotective function, making them useful in the natural sunscreen. Let's move to the problem segment. So MAAs are known to have the potential to be photoprotective and antioxidant. So due to the limitations of low accumulations of MAAs in organism, the cumbersome extraction process, difficult identifications, and high cost, MAA have not yet been widely used in human life. Due to the lack of understanding of the biosynthetic pathways, limited bioengineering approaches, and sustainable production strategies for MAAs, it has restricted the potential of the MAAs. Next, for the research question. First, what are the biosynthetic pathways and mechanisms currently used to produce MAAs? Second one, what are the bioengineering strategies that have been developed to increase the synthesis and yield of MAAs? And the third one, how can the current biosynthesis and bioengineering strategies of MAAs support the development of sustainable production methods? Next, for the objective. First one, to identify and compile published studies related to the biosynthesis pathways involved in the productions of MAAs. Second one, to analyze the bioengineering strategies utilized to enhance the synthesis and yield of MAAs. And the last one, to evaluate the current biosynthesis and bioengineering strategies support sustainable production approaches of MAAs. So, what is the significance of study for this systematic review? So, this review addressed the gaps in understanding the biosynthetic pathways, bioengineering, and sustainable productions of microsporin like amino acids. So, this systematic review proposed a comprehensive outlook that can be used for future research, for the industrial applications, and to promote the development of MAAs as a safer and more sustainable substitute for the traditional UV filtering compounds in other fields. So by systematically reviewing the current research, this can provide a true overview of MAAs productions. Let's move into the literature review part. So first, for the background of MAAs, according to Garrett et al. 2025, MAAs are a class of natural UV absorbing compounds with a broad spectrum of biological functions in cyanobacteria and algae. So certain photosynthetic organisms such as these cyanobacteria and algae have evolved to cope with exposure to UVR by producing MAAs. So next, what are the properties of MAAs? So according to Garrett and Pinto 2021 study shows that the MAAs are low molecular weight with generally less than 400 Dalton, colorless and water-soluble compound, and under environmental circumstances, they are extremely stable molecules. So according to Wada et al. 2015, the MAAs absorption maximum between 268 and 362 nanometer depend on their molecular structures. And last, cyanobacteria, algae, fungi, and some of the marine mammals contain MAAs. From Peng et al. 2023 stated that MAAs have been identified around 572 marine microalgae. Here we can see from this slide two types of MAAs, which are aminocyclohexanamine type and aminocyclohexanone type of microsporin like amino acids. Next, for the biosynthetic pathways of MAAs, so by Rosick and Duff 2011, the biosynthesis of MAAs is presumed to occur during the first part of the Shikimate pathways. So this pathway is responsible for the productions of essential aromatic amino acids such as tyrosine and tryptophan. So on the other hand, according to Chen et al. 2021, the pentose phosphate pathway is not the primary mechanism in the biosynthesis of MAAs. So already it merely serves as a backup to guarantee the diversifications of MAAs created due to the Shikimate pathway. Nonetheless, according to Garrett and Pinto 2021, there is a clear link between the both pathways. According to Mahagani et al. 2022, it stated that the genes coding for the mice cluster in the pathways were identified and characterized in silico approach. Here, the figure shows the biosynthetic pathways for both Shikimate and pentose phosphate pathways. Next, let's talk about the bioengineering approaches. So, to increase the synthesis of MAAs in many marine organisms, according to Ng Resami et al. 2023, gene overexpression is one one of the crucial genetic engineering methods. Meanwhile, according to Katosh et al. 2016, by transferring biosynthetic genes from native cyanobacteria or algae into more effective microbial hosts, heterologous expression is another important bioengineering technique. According to Rush et al. 2021, CRISPR is another way which also utilized for modifying the biosynthetic pathways to boost the MAAs production. Last, we move to the sustainable productions of MAAs part in this literature review. There are a few cultivation approaches which from Kaltekar et al. 2019. The first natural cultivation approaches are stress induction method under the UV radiations and light spectra. And furthermore, from Rush et al. 2021, it also stated that nutrient manipulations can also influence the natural cultivation approaches for the MA. For the methodology part, first, study design was conducted where using the PRISMA 2020 framework, which provide accessibility for existing approaches, patterns, and gaps. So next method is the database search. So the database search was conducted using this database, which are selected due to their comprehensive coverage in the related fields. In addition, inclusion criteria and exclusion criteria methods were also included in this methodology, where inclusion criteria, the articles were included in the review, whereas for the exclusion criteria, they were excluded and screening, which can be seen in the PRISMA 2020 flow diagram. And next, for screening and selections method were conducted, which the result were presented using the PRISMA 2020 flow diagram. Based on the flow diagram here, we can see the total of 33 articles were identified from the database searches. During screening, 14 review articles were excluded as they did not meet the inclusion criteria, whereas after eligibility assessment, a total of 70 full-text research articles were included for this systematic review. Next, data extraction were conducted, which to ensure the consistency and comparability among the included articles. Next, data synthesis were also conducted, which to facilitate structured comparison across the included articles. And lastly, these are the tools that were used, which are Microsoft Excel, Microsoft Word, and reference management software, which help to produce a systematic and reproducible review process. Next, for the expected outcome. So despite extensive research exploring the potentials of MAEs regarding biosynthesis and bioengineering, there is still a limited understanding of the complete biosynthetic pathways, bioengineering such as the genetic regulation and also integration on sustainability. So most of the studies just focus on a certain field of MAEs, and therefore a systematic review is necessary to emphasize the current knowledge gaps and emphasize the existing knowledge gap in the productions of mycosporin-like amino acids. Next, for the challenges part for this systematic review, there are several challenges, which the first one, the numbers of eligible studies is limited, which only studies that met the inclusion criteria were selected. Second, there is a limited data on industrial scales production, since most of the studies focus on the laboratory scale experiment. And lastly, this review is limited by database and language restriction, which has only selected databases and English language articles were included. Lastly, this is the Gantt chart that shows the timeline of my FYP activities. Next, this is the reference list for this presentation.